Okay. Oh, oh my God. That was a wake-up call. Yeah. So I think we start again. Our next speaker is Camila Canepa from the Universidad de Buenos Aires, Argentina. And she will talk about alpha interferon-loaded chitosan nanoparticles for the optimization of its therapeutic use.
Everything should work, also the point. Okay, great. And you have to Okay. Well, good afternoon. My name is Camila. I'm a PhD student from Argentina. And the work I'm presenting today is entitled Alpha Interferon-Loaded Chitosan Nanoparticles for the Optimization of its Therapeutic Use. So, type 1 interferon, including both alpha and beta, is a cytokine. Its direct action is to inhibit viral replication, but it also has an immunomodulatory action. So based on these two, interferon alpha 2b is nowadays approved for the treatment of some chronic viral infections and many cancer diseases. However, its short half-life causes to due to its short half-life, it has to be administered 3 times a week. Besides, its short therapeutic index and fluctuations in plasma concentration leads to serious adverse effects. So the objective of this work was to design and characterize a drug delivery system, or DDS, able to protect and control the release of interferon alpha 2b in order to diminish frequency of administration and related adverse effects. So we used chitosan, a cationic polymer. Nanoparticles were prepared with a ionotropic chelation method, and tripolyphosphate, or TPP, was used as an anionic cross-linker. So the interaction between these two was determined by infrared spectroscopy. A bimodal size distribution was observed by DLS results, and this was then confirmed by TEM imaging. A zeta potential of 31 millivolts was observed by DLS, suggesting physical stability, which was then confirmed by DLS measurements during 30 days. The encapsulation efficiency was of 96%, and as it can be seen here, we evaluated the antiviral activity of the loaded drug to the commercial one. And the loaded drug showed retained antiviral activity. So to sum up, we describe here a DDS that showed high encapsulation efficiency, stability, and retained cargo activity. So the next step is to define both in vitro and in vivo release profile in order to estimate the potentiality of this DDS in the optimization of the therapeutic use of interferon. And regarding the scope, this could impact on the quality of life of many people. And moreover, they described a simple method of preparation in mild conditions, aiming to preserve both structure and function of protein drugs, is also non-expensive, being suitable for the encapsulation of drugs of similar structure, such as interferon beta, which is now used for the treatment of multiple sclerosis. So thank you for your time.
Thank you very much. The next presentation is Constantin Mihailu Caciu from the University of Medicine and Pharmacy in Cluj-Napoca in Romania. He will talk about magnetic and plasmonic nanoparticles charged liposomes for drug delivery applications.
You are forward and backward and the pointer.
Okay, thank you. Thank you for the introduction. I will make a short overview of our recent results in the group of bionanoscopy in our university. After a few years ago, we started to work in this field of nanotechnology. We succeed to synthesize noble metal particles, nanoparticles, in classical way. But afterwards, we tried to make them more biocompatible, and therefore we used polyethylene glycol in order to make them more suitable for biological application. And finally, we try to combine these nanoparticles with other nanostructures like liposomes. In the first step, I will show you some data regarding how we succeed to form plasmonic liposomes. We used cationic lipids combined with phosphatidylcholine. 1:2 ratio was apparently the best ratio. And after checking, as you can see here are the zeta potential values for the liposomes, for the nanoparticles which are negatively charged. And after adding the liposome suspensions to the nanoparticles, the complexes which are formed still retain a positive charge, allowing them to be separated. These data were confirmed also by DLS data. So the particles, the liposomes, naked liposomes if you want, and afterwards, after they associated by electrostatic interaction, we do not have a significant change in the medium size, but an increase in the polydispersivity of the samples. And you have also here some time images. As you can see also in the UV spectra, we can notice a redshift of the plasmonic peak of the gold nanoparticles as they associate and form hotspots at the surface of the liposomes. This led to some interesting applications of these particles. A part of increasing the enhancement in the surface enhanced Raman spectroscopy, we also were able to notice that in the case of the pegylated gold nanoparticles, they enhance the vibration bands of the phospholipids. So eventually these gold nanoparticles coated with polyethylene glycol can be used as tools in order to check the surface of cells in the future. This was not possible to identify in the case of citrate-capped gold nanoparticles. So it seems that the presence of polyethylene glycol on the surface of the nanoparticle allows a better penetration into at the surface of the liposomes. Further, we tried to obtain also magnetoliposomes. In fact, we have also obtained some results with synthesis of nanoparticles in polyethylene glycol with a very good specific absorption rate, more than 2,000 watts per gram. But we wanted to have some mixed properties of these liposomes so that we can control their movement and also to be able, by using an external magnetic field, to trigger the release of eventually trapped substance. And you have some images, microscopic images. Also, nanoparticles were loaded with some drugs, and I'm sorry, I don't want to take your time. And also after disruption with detergent or other chemical substances, we were able to release the content of the liposome. So this is the idea, that we want to build a system which is able to load a substance and to have a control of the release of the substance. For other details, we have poster number 42. Thank you.
Thank you very much. Our next speaker is Jean-Baptiste Coty from Université Paris-Sud, France. And he's talking about the development of a multi-crossed micro-immunoelectrophoresis and application to the determination of complement activation at 50% by nanoparticles.
Thank you. I'm a PhD in the University Paris-Sud under the supervision of Christine Vautier, and I'm going to talk about the development of a high-throughput method for the assessment of complement activation by nanomedicines. So as you know, when we synthesize nanomedicines, we have a synthetic identity at the beginning, but for those who are destined to be administered by intravenous, they will acquire a new identity that is the biological identity through the formation of the protein corona. And this identity will provide new properties, and there will be interaction with the immune systems present in the blood circulation. So it will impact on the biodistribution of the nanomedicine and on its possible toxicity. And one of the main effectors of the immune response in the bloodstream is the complement system. So briefly, it's a group of 35 proteins that are arranged into a cascade of cleavage. And the protein at the heart of the cascade is the C3 protein that is cleaved when the foreign agent is recognized. So we decided to study the cleavage of this protein in order to assess the complement activation given by a nanoparticle, and that can give an idea on the stealth property of a nanomedicine. So we developed an immunoelectrophoresis method. So actually, there was already a classical method that was used. So in the first time, we incubate the nanomedicine with serum in order to observe the cleavage or not of the C3 protein. And in the method, we first do the migration in the first dimension on the agarose gel in order to separate the native C3 protein from the formed fragments. And in the second dimension, we make precipitation of this protein in the gel containing the antibody anti-C3. So with this first method, it was very long, more than 19 hours of electrophoresis, and we could only analyze 5 samples at once and 1 gel per sample. So we decided to optimize this method with an original concept of serial migration. So you can see here, we have 7 migration in the first dimension, and then we have 5 lines for the second dimension. So with the gel, we can analyze 35 samples at once, and we have a direct vision of the complement activation. So you see that we reduced the time of electrophoresis, and we greatly increased the number of analyses. Also, we validated the robustness of this method by ANOVA, and we were able to use it for a new application. Briefly, what we do is that we incubate different concentration of the nanomedicine, and we monitored the percentage of C3 activation, and we could have this kind of curve, and we could determine the surface needed of nanomedicine that was needed to cleave 50% of the C3 protein. So we did it for 3 model nanoparticles, and we see that we can have difference. For more stealth nanoparticle, we never reach the 50% of activation. So this parameter that we call the C3A50 could become a parameter that can reflect the behavior of nanomedicines toward the complement system. So if you want more information about it, you can come to poster 97. Thank you.
Thank you very much. Our next microphone. Now, okay. Thank you. Our next speaker is Nathalie Pinkerton from CNRS Toulouse in France. She will talk about LANCER, light-activated nanocarrier release.
Does it point? Yeah. Perfect. All right. Well, good afternoon, everyone. I'm Nathalie Pinkerton, and I'm from Toulouse, a lovely city in the south of France. And there I'm a postdoc at ITAV. There we go, much better. There I'm a postdoc at ITAV, an interdisciplinary CNRS institute, which is focused on basic and translational biomedical research. So there I am part of the SOMO chemistry group, which is led by Dr. Stéphane Chassin, where we're interested in developing chemical tools for organic synthesis and also for diagnostic and therapeutic applications. So I lead the second subset of the group, and today I'd like to introduce you to one of our main projects entitled LANCER for light-activated nanocarrier release for controlled drug delivery. So like many of you here, we are interested in addressing several of the challenges associated with chemotherapy, in particular the limited water solubility of drugs, and perhaps more importantly, the systemic delivery of cytotoxic drugs, which results in several unwanted side effects. So in order to address these challenges, we are developing a tumor-targeting nanocarrier that, upon light activation, releases anti-cancer agents in a dose-dependent manner. And so our approach is quite simple. We are developing a light-responsive platform to which we can couple a variety of drug molecules to create a light-responsive prodrug. And then, once we've created the prodrug, we encapsulate these prodrug molecules into a prodrug-loaded nanocarrier, shown here, via a controlled precipitation method called flash nanoprecipitation. And this enables the formation of highly pegylated nanocarriers, which then preferentially accumulate within the tumor environment. So from there, we selectively illuminate the tumor environment to only release the drug molecule within the tumor environment to give us spatial specificity and also control over the temporal release. So in addition to the drug release, we also form a fluorescent reporter, which then gives us confirmation that we've actually released the drug molecule in the location and in the dose that we desire. So just to give you a small taste of our results, this is our proof of concept platform here. Our light responsive moiety is based on the orthohydroxycinnamyl group here, to which we've conjugated N-diacetylcolchicine. This is a tubulin polymerization inhibitor, and together we create this light responsive prodrug. When it's illuminated with light, it releases the drug N-diacetylcolchicine and forms this coumarin fluorescent reporter. So we've successfully synthesized this molecule and encapsulated it within pegylated nanocarriers. These nanocarriers are able to controllably release the drug as a function of light exposure. And in vitro shows some very interesting results. So as I said, N-diacetylcolchicine is a tubulin polymerization inhibitor, so it greatly affects mitosis. So in these alpha-tubulin GFP HeLa cells, when we incubate the nanocarrier with the cells but do not expose them to light, we observe normal mitosis. However, when we illuminate the cells, we release the N-diacetylcolchicine from the nanoparticle, and then we actually create abnormal mitosis and eventually kill the cells. So please come say hi at the poster if you'd like more information on this project or any of the other projects in our group. Thank you for your attention.
Thank you very much. Our next speaker is Vivek Handra. He's an independent researcher in India, and he will talk about attributes affecting site-specific nanotherapeutics.
Thanks, Dr. Schmidt. Good afternoon, everybody. We've been having brilliant sessions since the morning, so I'm not going to take any time of yours, just 3 important points, and all of them are really relatively groundbreaking and new as in terms of concept physics in nanomedicine both. The first thing is I will be talking about nanorams. These are nano entities capable of sensing, signaling, and information processing, and thus we can achieve ultra-specified localized drug delivery and also defense system like immunity enhancement and stuff. And the target would be to cure diseases rather than to manage what we are presently doing. I'm more from the clinics than from the technology part of the nanomedicine entity. So, and reduction in global treatment cost is that is what is one of the main targets in our group. So, nanorobotic assistance machines, what we call as nanorams, they two things which are important to mention today. One is the light up signaling method. And the second one is the enzyme turn off mode. So first, the light up signaling method. Now, light up signaling, what we are targeting to do is we inject a bleb of the nanorams in a type 2 diabetic patient. And as the sugar level fluctuates, there will be a radiation of light, so luminescence from the bleb. So if the patient's got unexpected hypoglycemia, so we can watch from outside, and rather than having a glucometer to test it quickly, you can just see the alarm light and then react. And if he's got unexpectedly low sugar levels, you can take the concerned action. And the enzyme turn-off method, that's more inside the body, where we are targeting to touch the gut enzymes like DPP4 or the kidney enzymes like SGLT2. Both in we're targeting to get hold of the glycemic levels in diabetic patients again. And so what I'd like to emphasize here is that there is a need of more extensive global collaborative endeavors from all across the globe so that we can actually accelerate the domain of hybrid technologies wherein medicine and nanorobotics are almost on the same page there. And that calls for a small upgrade, maybe a nano upgrade. Thanks very much. Thanks for your time.
Thank you very much. The next speaker is Deborah Bonvin from the École Polytechnique Fédérale de Lausanne in Switzerland, and she will talk about small and biocompatible coatings of iron oxide nanoparticles for improved detection and treatment of lymph node metastases.
Thank you very much. So it's really my great pleasure to give you an overview of one part of my PhD thesis, which focused mainly on the coating of iron oxide nanoparticles for one specific application, which is the treatment and the detection of lymph node metastasis. So I don't need to explain you that iron oxide nanoparticles are very promising for medical applications. But one important point is that in all these medical applications, they are all in physiological conditions, which means pH around 7.4. So this means, and if you look at the surface potential in function of pH, they don't have, let's say they don't have any charges around at this physiological pH, so they are not stable. So we have to coat them in order to stabilize them. There are two main strategies for that. The first one is the steric stabilization to provide a distance between the nanoparticles, and the second one is electrostatic stabilization by charges. Usually people use the first strategy with very large molecules such as polymers, sugars, etc. But this is not the strategy that we decided to go for. I told you at the beginning, we want to target lymph node metastases to treat them by hypothermia and to detect them by MRI. So we decided to go for rather small molecules, small charged molecules, in order to have a very thin coating. Why? Simply because if you have a thin coating, you have a small nanomaterial at the end. So you can pack much more nanoparticles in this very small metastasis, which means that you will also have higher heating for hypothermia and higher contrast for MRI. I would like to show you one of the examples that we use, which is this folic acid. It's a vitamin that you already have in your body. And what we present in the poster is first the physicochemical properties of the nanoparticles, of the coated nanoparticles with this folic acid. So we show that we have a size range around 80 nanometer. We have a negative surface charge, which was in fact shown to be good for lymphatic access and retention. So if we want to target lymph node metastasis, this is, let's say, the optimal system. We have a very small and thin layer around the particles. We show that we have high relaxivity compared to a commercial contrast agent. We have very high heating for the hypothermia. In vitro on cells, we show very low toxicities and very specific uptake. If you want to know more, you can come to my poster. It will be a pleasure to explain you a little bit more. In a broader perspective, so what we want to do with the same product, theranostics, which means diagnosis and therapy. And this is very beneficial for the patient. We hope to be able to detect lymph node metastasis earlier than what can be done at the moment, which would really save lives, and at the same time to treat these metastases. Maybe one more point. One problem that we have at the moment is the targeting. It was mentioned today already a few times, how to bring the nanoparticles to the tumor, and this is one of the challenges that we have at the moment. Thank you very much.
Thank you. Thank you very much. Our next speaker is Shima Tawakol from the Drug Nanocarriers Research Core in Iran, University of Medical Sciences, Tehran, Iran. And she will talk about an investigation on the spinal cord recovery potential of self-assembling peptide nanofibers in vitro and in vivo.
Good afternoon, ladies and gentlemen. I am assistant professor at Iran University of Medical Science and head of non-drug delivery research course.
You have to point in this direction. Yeah, now, okay.
Today, spinal cord injury as an incurable disaster has remained and that is associated to paralysis. As seen in the figures, a spinal cord injury defined as a loss of axonal integrity. Despite of many studies in the field of neurodegenerative disorders, but thus far, a successful recovery of spinal cord injury has been achieved. It is estimated that about more than 300,000 patients struggling with STI in the United States, and this number are increasing. Science implantation of perform scaffolds in the site of a spinal cord injury or a brain, a soft tissue, induce higher inflammation and higher intracellular scarring in the site of injury, and not filling of void space in the site. Use of and implantation of scaffold in situ scaffolds with hydrogel-based structure has been preferred. In our investigation, we choose 4 groups as a self-assembling peptide nanofiber. At the first was the matrigel as a thermogel nanofiber as a positive control, and the next was RADA or with third name of Puro Matrix as a core of self-assembling peptide nanofibers, and third and fourth was belong to the core of self-assembling peptide nanofibers via 2 glycine anchorage that bonded to the long motif laminin that its sequence is patented in Iran, and the fourth was a core of self-assembling peptide nanofiber via 2 glycine anchorage that it is bonded to the bone marrow homing peptides. Then cells, neuroblastoma cells and endometrial-derived stromal cells encapsulated into the nanofiber, and cell viability, protein, and gene expression related to neurogenesis has been investigated. Results show that neuroblastoma cells, cell viability of neuroblastoma cells was in good agreement with the BCL2 gene expression, while TUG1 gene expression was in good agreement with the cell viability of endometrial neuron-like cells derived from endometrial stromal cells. Science osteoglomerular scarring, GFAP expression, a marker of reactive osteocyte is a major markers for the inhibitor of recovery of spinal cord. So GFAP gene expression has been investigated. Results show that third of self-assembling peptide nanofibers suppress GFAP gene expression, while material as a control group express GFAP, induced expression of GFAP. Protein expression data conferred of gene expression. And then GABA and tyrosine hydroxylase genes were investigated. Our results show that tyrosine hydroxylase has overexpressed just in the cells encapsulated to laminin, long motif of laminin. This nanofiber has selected for further investigation. Tubulin polymerization and depolymerization via circular decrease method show that in the concentration that is parallel to the tubulin, we have similar behavior such as polymerization and depolymerization of tubulin, but less or higher concentration of self-assembling peptide nanofiber induce suppression of the decrease or suppress the polymerization and polymerization of tubulin. Then we continue our investigation via in vivo study. The self-assembling peptide nanofibers implanted to the SCI model of rats, and then myelin synthesis, neurofilament overexpression, and dark neuron has been investigated. Results show that although although long motif of laminin in the in vitro show that higher neurogenesis as compared to bone marrow homing peptides, but bone marrow homing peptides motifs connected to core self-assembling peptide nanofibers induce higher myelin deposition and decrease of GFAP gene expression and protein gene expression, and induce less level of dark neurons in the tissue specimen. We can make, and after that, BBB score showed that and then BBB score showed that bone marrow homing peptides induce higher level of motor neuron recovery as compared to the others. In conclusion, it may be said that self-assembling peptide nanofibers with definitive biological motif can and chemical-mechanical transduction signals are promising for the motor neuron recovery. Our future study is on the encapsulating of some drug as an anti-inflammatory small molecules into the self-assembling peptide nanofibers with help of electronic muscle stimulator. Thanks for your attention.
Thank you very much. Our next speaker is Gesine Heuk from Precision Nanosystems in Canada, and she will talk about production of size-tunable PLGA nanoparticle drug delivery systems using microfluidic technology.
Very well, thank you. Thanks for the very nice introduction. I would like to present actually just one poster. Which is the 108, but we actually have two posters here. This 108 one is on the PLGA nanoparticles, but our company actually owns a microfluidic platform that can be used for a number of types of nanoparticles, including and especially liposomes and lipid nanoparticles. But these PLGA nanoparticles are a new application for those who already know our platform. And that's why I'm presenting this here today. So why do we actually want to make polymeric nanoparticles with a microfluidic platform? If we look at the development phase of these nanomedicines, we still have a few problems. There's inability to achieve particles with a size below 100 nanometers. It's very hard to fine-tune the particles. It's very hard to fine-tune the particle size, to fine-tune the particle polydispersity. There's a lot of batch-to-batch variability. It's hard to reproduce because it's very dependent on the person who makes the nanoparticles. Well, the outcome is very dependent on the person who makes the nanoparticles, and it's hard to transfer protocol from one person to another because a lot of expertise is needed. It's difficult to scale up. And one point that's not mentioned here is also that if I want to load a polymeric nanoparticle with the conventional methods, an empty nanoparticle has a specific size, and as soon as I add a drug, suddenly I end up with a polymeric nanoparticle of a larger size. So it's hard to compare the control with the new nanoparticle, with the loaded nanoparticle. So we want to try and allow you to compare apples with apples and not apples with oranges. Now the microfluidic solution, I'm already late. The microfluidic solution is that we have a microfluidic cartridge here where we inject the PLGA dissolved in acetonitrile here, and here we have an aqueous solution with a stabilizer, which is polyvinyl alcohol. We just mix it, and this mixing platform is really key to getting control over the nanoparticles. It's very fast, and it's in a very, very confined volume. We mix within milliseconds, and we mix in a very small volume of a few nanoliters, which is not what you find in conventional methods. And these are just a few results that I want to present. So in this microfluidic chip, we can change the ratio of the two fluids to another, but we can also change the total flow rate with which we inject two fluids into the cartridge. And my colleague, Sham Garg, he played around with this, and he injected the organic phase and the aqueous phase at different speed. And by that, he can actually control, he and now you and I, we can all control the particle size just by using different instrument parameters. And it's not the only thing that allows us to control the size. We can also just change the polymer concentration in the organic phase. Knowing that these results are all reproducible, we can now control the particle size just by changing the concentration of the PLGA in the organic phase. Sham also did a test with a model drug, coumarin, which is a hydrophobic drug. And he was very pleased with these results. He was able to reproducibly, at different instrument parameters, he was able to load coumarin into polymer, the PLGA nanoparticles of up to 70%, which is quite a bit higher than what you see in the literature with conventional methods. And this is the key graph, I won't be long, where you can actually see that he loaded these nanoparticles and he was able to keep the particle size the same. So now his control particles and his loaded particles are the same size, same characteristics, so he can now compare apples with apples and not apples with oranges. That is key when you work in vivo, right? Because different particle sizes land in different tissues. To wrap up, I was supposed to answer, or we were supposed to answer the question, how does this actually fit into nanomedicines? So if you have better size control or better control over how the particle size is, if you have very good reproducibility, and if you can use an automated platform, which the NanoAssembler platform is, that is scalable. I did not speak about it, but please come to our booth. I am happy to talk more about it. You can improve rational design of your particles in vitro and in vivo. You will see, once you see how fast the instrument is, it's very fast development of your nanoparticles. And above all that, it breaks down these massive hurdles we have in the development process of nanoparticles for the clinics. Yeah, I hope this wasn't too fast, but it was clear. And as I said, please come to our booth. It's right there near the coffee. And please also come and see our posters 108 and 109.
Thank you very much. Is my microphone on? Yes. Okay. Our last speaker is Angélique Sour from the Université du Strasbourg in France, and she will talk about theranostic agents for MRI and 1 or 2 photon PDT in vitro and cellular studies.
Thank you for your kind introduction. Good afternoon, everybody. I would like to thank the organizers for giving me the opportunity to present the work done in Strasbourg. We are developing theranostic agents for magnetic resonance imaging, MRI, and for photodynamic therapy, PDT. Photodynamic therapy requires a photosensitizer, light, a light source, and oxygen. The photosensitizer is administered intravenously into a patient. After several hours, it accumulates into the tumor, and it is then irradiated. To improve, this irradiation is absorbed by the photosensitizer, and the porphyrin produces oxygenated species which induce cell death and also tissue death. To improve the efficacy of this technique, we can also choose to irradiate in the near infrared, which corresponds to the optical transparency of the tissues. And an efficient way to reach this near-infrared region is to use the 2-photon absorption process. The 2-photon absorption will allow deeper treatment and also the treatment in a much more narrow region. We then attach to the photosensitizer a gadolinium complex, which will bring the imaging modality. MRI is a powerful imaging technique thanks to its excellent spatial and temporal resolution, but it suffers from a weak sensitivity. And a way to increase the sensitivity is to design medium-sized molecules and also relatively rigid molecules. You can see here an example of a theranostic agent. Okay. On the left, you can see the porphyrin derivative decorated with triethylene glycol chains, and this will behave as the photosensitizer, and it is linked to a gadolinium DOTA complex, which brings the imaging modality. This compound has been studied in vitro in water, and it has the potential to behave as an efficient contrast agent for MRI, thanks to its relatively high relaxivities over a broad magnetic field. It has also the potential to behave as an efficient photosensitizer after 1 photon and 2 photon irradiation. Finally, it is also able to produce singlet oxygen. We then performed cellular studies. You can see here images which correspond to the fluorescence of the molecules, and on the bottom, these are phantom MRI images. They also show the internalization of the compound inside the cell, and they also tell us that the gadolinium uptake is 16 times higher than the commercial contrast agent. And this can be explained by the presence of the lipophilic porphyrin part. We then also performed the phototoxicity studies, and we could show that this compound is able to kill the cell after 1 photon and 2 photon irradiation. And this molecule brings the first proof of concept of a potential theranostic agent for 2-photon PDT and magnetic resonance detection. Finally, in order to increase the amount of theranostic agent into a site of interest, we develop the theranostic agent with targeting units. And we linked the photosensitizer to folic acid. We also recently collaborate with a group and succeeded to incorporate a theranostic agent into a cubosome. I would like to thank you for your attention.
Thank you very much. And with this, I would like to conclude this session, and I would like to thank all the speakers once more for interesting and to-the-point presentations. They were very clear, all of you. Thank you very much.